Thank you very much indeed, Mark, and good morning, ladies and gentlemen. We all know from life that sometimes the most simple things in life are the ones that are most effective. What I'm going to tell you about is a very simple idea that was developed by Angle plc in 2005, and over the intervening period we've developed and patented this very, very simple idea, which is that you can capture cancer cells in the bloodstream of cancer patients by virtue of their larger size and lack of compressibility, and we can apply that in the personalised cancer care for every cancer patient in the world. So it's a very simple idea, which is owned now by Angle plc, which has a very wide applicability in the treatment of cancer patients. So the background to this simple idea is that obviously the incidence of cancer is growing very fast. Unfortunately, we all in this room have a 1 in 3 chance now of suffering from cancer, and it's moving inexorably towards 1 in 2. So it's crucially important to improve the treatment for our cancer patients. The characteristic of cancer, however, is that it continually evolves and mutates, and that means that each patient's cancer is very different from the next person's cancer. So that if two people have colon cancer, they will have different diseases, notwithstanding the fact that they've both got colon cancer. And this is crucially important in terms of treatment. A little known fact is that many of the cancer drugs that are administered to patients don't actually work for that patient. And in fact, in some cases, they not only don't work, but they give the patient a worse outcome than if they'd not been treated at all. And this is why patients are typically given multiple cancer drugs at the same time, because the doctors simply do not know which drug will be effective for which patient. Now, this is set to change with something called personalised cancer care. Really, to effectively give the patient the right treatment, you need to understand their individual cancer, which means looking at their cancer cells, taking the cells out of the cancer and analyse them in a laboratory to work out what's the mutational status for that particular patient. And you can see at the bottom there are a number of targeted drugs which are being developed, and these ones, the 3 at the bottom, are already on the market. Herceptin for breast cancer is the best known one of those, but these are already on the market and they're being targeted to patients based on the results of their biopsy. So a biopsy is, if somebody has breast cancer, they cut the lump out and they take the cells from that cancerous lump and investigate them. Now, this is the whole essence of what we're doing with Angle. So the thing about biopsies is that it's quite difficult to do. It's an invasive procedure. It's a surgical cutting out of material. And some cancers it's quite difficult to access. So for example, imagine somebody's got brain cancer. You don't want to cut into their brain unless it's actually part of their treatment. To do that just to find the cells is something you wouldn't really want to do. And so some cancers are difficult to access for biopsy. Regardless of that, the cancers mutate over time and they change. So that they do an investigation of biopsy right at the beginning of the treatment and they find out the mutational status. 3 months later, 6 months later, when the patient's in treatment, the cancer's changed and they have no way of knowing how it's changed because they've got nothing to cut out. They already cut it out. So there's nothing left to surgically excise. The essence of what Angle is doing with its Parsortix system is what we're calling a liquid biopsy. So as an alternative to cutting out parts of the cancer, what we're doing is giving the patient, well, we're not giving it, the doctor will give the patient a simple blood test, just exactly a normal peripheral blood test. And inside that blood sample, just a small tube of blood, there will be a very small number of the patient's cancer cells, because that's how secondary cancers are created in different locations. The primary cancer drops cancer cells in the bloodstream, and they float around in the blood, circulate in the blood. They're known as circulating tumour cells. They land somewhere else, and that's what can cause a secondary cancer. Now, the thing about these cancer cells is there's only a very small number of them. Typically, there might be 1 cancer cell in 1,000 million blood cells, and that's why it's been absolutely incredibly difficult to get hold of these cells. However, if you could get hold of those cells, you have the ability to give the patient a repeatable, non-invasive blood test periodically, get the cancer cells out and work out which drug will help that patient. And that is what we are focused on in Angle. And if you come to our stand, which I think is stand number 9, but it's the first one you see as you come in from registration, right in front of you, you will see this machine here on display, and you will see this consumable. And that consumable is basically a simple piece of plastic, but inside there it's got a patented system based on a series of steps for capturing the cancer cells. Now we're very lucky that, in the sense of being able to do this because the cancer cells are about 2 or 3 times bigger than the white blood cells. So this is a form of filtration. We're capturing the cells because they're bigger and also they're less compressible. Cancer cells are not designed to be in the blood. Blood cells are designed to be very compressible so they can get through little tiny capillaries. But cancer cells aren't, so they're less compressible. And so in our system, they get caught on these patented step in here. So basically, that's the blood that's drawn straight out of the patient. We take the top off the tube, attach it to our machine. It's automated. It pushes the blood through a micro-fluidic system, through the cassette, which is sitting in here, consumable. And all the blood goes into here, which is the, which is the waste container. And the cancer cells are caught along here. Now, the beauty of this system, and this is where we stand out from a number of other companies who are trying to do something similar, is that we can actually reverse the flow and take them out and collect them. And we call that process harvesting. So all we're doing is providing the cancer cells in a different way. So instead of cutting them out, we strain them out, filter them out of the blood and provide that for analysis. And so it's quite a simple approach. Now, it's a simple approach which we've been working on for a number of years, as I explained, and we recently announced the results of Cancer Research UK's evaluation of our system over an 18-month period. And they have the Patterson Institute for Cancer Research up in Manchester. That is associated with the Christie Hospital, Europe's largest cancer hospital. They had a full-time senior technician working with our system for 18 months, and they've gone right the way through it, and they've helped us to improve it. And they've basically published information that shows that we have a very high level of capture of cells, between 80 and 100% of the cells, a very small number of cells that are there. And we have a high level of harvest, so we can get about 60 to 100% of the cancer cells out for analysis. I'm going to go back to this, but they're particularly pleased that we don't use antibody capture approach, which is the main alternative way of doing it. I'll explain that. We have a low level of white blood cell contamination, which means that the harvested cells are in a form that they can be analysed. We've got, here there's 3 major research partners. University of Surrey is our local university. Actually, the Royal Surrey County Hospital is a tertiary referral centre for cancer for the south of England. And they've been working with our system as well for about 18 months. We're also working with the Medical Research Council, which is obviously the government's arm, with the University of Cambridge and Addenbrooke. So we've got 3 world-class key opinion leaders. In fact, we announced this week, so we're expanding our key opinion leader programme. We announced this week that we have signed up Hamburg, which is the leading German circulating tumour cell group, and they're working with our system. So the way our system works is that we have a patented step-based system. The good thing about it is it captures a high number of the CTCs, and it works for all different types of cancer. The competing systems only work for a few cancers. It doesn't use these antibodies, and I won't go into all the detail there, but the thing about the antibody system is it's very complicated, it's expensive, and it doesn't capture all of the cancer cells that are of interest. The other alternative approach is something called the membrane systems, and that also is based on the size of the cells. However, the problem is they can't get the cells out for analysis. Right, so what are the commercial advantages? Because this is what business is about. We want to make a big difference in improving the treatment of cancer patients, but we want to do that in a financially effective way. We already have a CE mark, which is the clinical use authorization for basically using this, treating patients in Europe. And we're working on getting the FDA authorization for treating patients in the United States. And the last point there is probably absolutely crucial. It's open source. So if you remember the analogy with the computer companies, is that you can have something which is closed code or open code. Open code means it works with everybody's systems. It's not just a proprietary basis. And that's what we've got here. So the whole idea of our system is to take the cancer cells out, so the existing molecular platforms, which already sit in these pathology labs in the hospitals, can be used. So we want to work with the biggest companies in the world who are already doing molecular analysis of cancer cells. All we want to do is give them some more cells which they can't otherwise get. So that's our idea is that our Parsortix system should be compatible with every other system on the market. and therefore can be widely used. In terms of what we're actually doing now to secure our position, we're concentrating on rolling this out with other key opinion leaders. So I'm in the process of setting up arrangements with the major cancer research centres all over the world. So we want the biggest and the best cancer research centres in the world using our system and using it for a variety of different cancers. So we're setting up big studies. That's where you'll expect to see a lot of news from Angle over the next 12 months in particular, is setting up more key opinion leaders. Because those people need to show that if you get the cancer cells out of the bloodstream and you find a particular mutation in a particular type of cancer, then you give the patient a particular drug, it improves their outcome. So that's the piece that's missing for us. And we're addressing an £8 billion market. So we have a proprietary position with protected patents in an £8 billion market. And the gap between us and servicing that market is getting some of these cancer research centres saying, we use this with metastatic breast cancer patients. We chose to give them Herceptin when we wouldn't have done otherwise as a result of this intervention, and their treatment improved. That's what we want to do in all the different cancers. And so that's the summary point, is that we're getting clinical applications for how well the treatment can be with using these circulating tumour cells. And that plus the regulatory authorisation and the clinical application is what takes us into the $1 billion market. And just to sum up here, the absolutely core thing here is that the Cancer Research UK have said personalised medicine is the most exciting change in cancer treatment since the invention of chemotherapy. That is what we are working on. We are enabling that. We are providing the basic cells that they need in order to give that personalised cancer care. Same thing in the United States. This is the US government's major group. Personalised cancer care is the future of medicine. The faster we can develop cancer treatments that are tailored to the individual characteristics of each patient, the better we can concentrate interventions on those who will benefit most from them and avoid the expense, side effects and emotional involvement on those who will not. So you imagine a situation which is an absolutely real situation right now where a patient is given a drug which is costing £60,000 for a course, and 40% of the patients get no benefit from that. That's wasted £60,000 for those 40%. Worse than that, they suffered the toxic side effects of that drug. And in some cases, for example, in ovarian cancer, I saw a recent study, that it actually reduces the patient's long-term overall survival by 6 months. So that is an absolute terrible thing. It's a waste of money and it's bad for patients. What we're looking at is something that's very, very simple, it's low cost, and it enables them to make the right decision. So that patient will not be given the wrong drug, they'll be given the right drug. And this is also being driven by the big pharmaceutical companies. So they're all looking into how can we get new cancer drugs which are targeting particular mutations. So they're also another area of collaboration for us. We want to help provide them with the tools so that they can find out which are the drug targets for the future. And there's a big drive towards something called companion diagnostics, where new cancer drugs should have a diagnostic alongside it to identify which patients will benefit from this particular treatment, and then to show that they have benefited from it. And that is what we're playing into. So, and you can see here very summary comments from our research partners of how well this system is working. The reason I'm here today is because I want to get this message out. We've been working on this now for about 8 years. We're just actually now at the point of breakthrough. We're moving into the research market already, which is a £250 million market. And soon I hope to bring this to patients and improve the outcome for all those patients with cancer. Thank you very much indeed.